Um, we'll start the webinar, and this is the fifth one in this Cyclodextrin Masterclass. I'll switch this on. And today we are going to be a bit more technical. So during the former occasions, if you saw those events, we have been focusing mostly on applications, I would say, in different areas, small molecules, biologics, non-pharma applications. Today, we'll be, again, a bit more technical and discuss how we can make a cyclodextrin complex, what kind of technologies are out there, what are the options, how you can make sure if you want to make a complex that the complexation happens, and so on. As always, or as with the former events, I'll talk for about 45-ish minutes. At least that's how it's planned. In the meantime, you can ask all your questions in the chat box, or you can save them for the Q&A session that's gonna be at the end of the presentation. But eventually you have to raise them in a written form in the chat, and I'll try to tackle all of those that I can, at least in the last 15 minutes. Also, if you registered and cannot attend or have to leave or whatever, we'll distribute the recording. This is a recorded event. Distribute the recording of the webinar later to everyone who registered. So let's kick off. So what we'll talk about today, how to make a cyclodextrin complex. In case you're not familiar, this may be a bit boring to some of you, but we'll talk a little bit in general about cyclodextrins. Then within the complexation session, the core part of the presentation, I'll discuss three main areas: how to optimize a composition of a cyclodextrin complex, what kind of techniques are out there, industrial techniques to make cyclodextrin complexes on a commercial or large scale, and what kind of analytical tools do you have to make sure that the complexation did occur and to properly analyze the complex that you made, hopefully. So this is what we'll talk about today. Let's start with the general considerations. As you know, our favorite molecule, the cyclodextrin, has this nice truncated cone or donut shape. And of course, the key part is the cavity, which has a hydrophobic nature and can form inclusion complexes with a wide variety of molecules that's exploited in a wide range of industries, mostly used as excipients, of course, for a variety of uses. I'll talk about this later. Nowadays, we see more and more novel applications emerging, especially in the pharma field, for uses as active ingredients in different types of biologics and biotechnological processes. But traditionally, the complex formation is the key, and this is what we'll talk about today. So how does the complex formation occur theoretically on a molecular level? As you can see, we have a cyclodextrin here and an active ingredient. And of course, if this active ingredient is water repellent, let's put it like that, or doesn't enjoy being in water, it would certainly enjoy being in the cavity of the cyclodextrin because both are hydrophobic, so they attract each other. And if the active ingredient can sit in the cavity of the cyclodextrin, that's an environment that it prefers much more than the aqueous environment that's otherwise. And that's how the solubilization technically occurs. As you can see, this is not a one-way street, so this is an equilibrium. This kind of dissociation and association happens all the time. So the complex forms and it dissociates. It forms and it dissociates. And in order to exploit the best characteristics of the cyclodextrins, you have to make sure that this equilibrium is shifted as much as possible towards complex formation, at least when you want the active ingredient complex. And of course, in order to exert its effect, you want it to be released from the cyclodextrin cavity, which you can also tune in a way. So what happens when when the complex is formed. You can see a lot of immediate advantages of such. So technically what happens is that the cyclodextrin, a little bit as visible in this picture, would mask the presence of the active ingredient, and accordingly it would mask all its properties. So this means that it would mask its taste, it would mask its smell, it could mask the irritation it would cause on the surface of the skin. So to some extent it could mask the toxicity itself. And also it can increase the solubility, because this complex has usually the solubility of the cyclodextrin, not the solubility of the active ingredient. Not always, but okay, we'll get to that. It can improve the stability, the wettability, the bioavailability, and so on. So we have a lot of advantages that we can talk about. You'll see that when we're talking about cyclodextrins, we're not talking about a single molecule. So they come in different sizes and in different modifications. And why the modification was important of the cyclodextrins. So what we're talking about is that to modify the cyclodextrins in this position and this position and this position. So the OH groups of the cyclodextrin can be chemically modified in order to improve the solubility of the cyclodextrin. Dominantly, that was the original idea, in order to improve its safety profile, and eventually because its functional properties are much better that way. So it can interact with the guest molecules on a wider range of secondary interactions. So stronger complexes can be formed, and accordingly modified cyclodextrins usually outperform the native ones. Not always, but usually. So, a little question for you. So, I don't know how much you know about cyclodextrins. What's your guess? How many cyclodextrins are used currently in pharma products? You can type your guesses in the chat if you wish. So, altogether, with all the applications that are out there, let's try to figure out how many cyclodextrins can be used in pharmaceutical products currently. Okay, I'll leave you just 5 seconds to think, and then I'll move to the answer. It's a bit surprising because this number is fairly high, I think, in my opinion. So 9 cyclodextrins are used in pharmaceutical formulations. 7 are used for small molecules, typically as excipients, stability improvers, whatever the typical uses. 1 is specific for biologics as a vaccine adjuvant, and 1 is approved as an active ingredient. And I'm quite sure that this number will grow in the coming, 5 to 10 years in pharma. It takes time, but we're getting there. So what kind of uses out there for cyclodextrins? Just apart from solubilization, you can use them for taste masking to ensure content uniformity, especially for highly potent drugs. So technically as a filler, similar to other oligosaccharides, but with further added values. You can improve the dissolution, especially if you want a very quick effect and a fast onset for something like a painkiller or against gastric acid or something like that. It's completely feasible with cyclodextrins, chemical stabilization, as we mentioned. And in a lot of cases, it's part of the life cycle management process because cyclodextrin complexes are usually patentable, although the literature is quite waste, but but if you can find some niche areas, it's well patentable. Just to mention, its compatibility with different kind of dosage forms. Technically, there are no dosage forms that technologically prevent the use of cyclodextrins. So you can see that there are solids, semi-solids, liquid formulations, wipes, sprays, chewing gums. Technically, you can incorporate them into any kind of formulation. They are really friendly in that way. Why would you use cyclodextrins? Just to bring you a few examples from the market, what it means, what kind of improvements you can expect from cyclodextrins. So the example of alpha-oxalone, Here you can see the dramatic change of the aqueous environment that was needed in order to solubilize 10 milligrams of the API, so that a final product could be created. This is a veterinary product of alphoxolone, by the way. Or you can improve the bioavailability rather dramatically. This is a glucagon-based nasal spray from Eli Lilly, where I don't know, 7-fold bioavailability improvement was achieved with the help of cyclodextrins. Or you can increase the shelf life, as you can see, from a few weeks to 2 years. And the beautiful part is that these kind of effects you don't just achieve alone, but they all come combined. So of course you will increase the solubility, but that will come with a bioavailability improvement. And if your active ingredient is sensitive and can degrade fast, it can easily come with a stability improvement. So I don't know, in case of itraconazole, the oral solution, the dominant primary goal of using cyclodextrin was solubility improvement, but it also influenced the shelf life. Similar examples can be brought from the nutraceutical area, of course, based on the very same mechanism of action. Some of these slides we brought from Walker, so you can see how much it improves the bioavailability of coenzyme Q10. You can use the cyclodextrins to make oily materials into a powder form with their help, similarly to silicas, or just to make I don't know, the absorption of certain components much better. Again, some data from Walker. So a lot of uses, but only if you really achieve a true complexation. And this is the critical part, and I really hope that I can explain it in a way that's understandable to everyone else, because this is a very key component of developing a good product. We often see products on the market when we analyze them, where among the ingredients, and this is not really true for pharmaceuticals, but often happens with nutraceutical applications actually, that we see the cyclodextrin among the components of the formulation, but when we analyze it, we realize that there is no true complex formed. And that's a problem, as you will see, because making the complex is not utterly complicated, but if you miss to make the complex, you miss to exploit the potential of the cyclodextrins. So this is why. This is the example of remdesivir, which was the first approved drug against COVID, you may remember, and it was solubilized with the cyclodextrin. So if you just take the composition that was approved, put the huge amount of cyclodextrin and the very small amount of active ingredient, and do a physical mixing, this is what happens. So of course you can create a powder, but this powder doesn't get dissolved. The cyclodextrin part gets dissolved. This is what you see, but the active ingredient doesn't get dissolved at all. And that's because in a physical mixing, complex formation doesn't occur, or only to a very little extent, depending on what kind of procedure you use. So the API doesn't get into the cavity, or doesn't get into the cavity of the cyclodextrin, and accordingly its properties are not matched, so it doesn't become water soluble. If you use the exact composition and create a lyophilized sample after proper processing, you can see that the same composition is water soluble. If you use another technology like electrospinning, you create a higher surface and you can dissolve the stuff even faster. So let's see, and I know this is a bit too much, but don't be afraid. We'll break it down to 2 slides in the. So this is technically the essence of what we'll talk about in this lecture. What kind of technologies out there? What kind of solubilization tools we have? and how can we use cyclodextrin? So obviously, solubility is a big issue still in the pharmaceutical industry, and probably it will become, at least in the small molecule space, a bigger and bigger issue as we invent novel actives, because these are the trends. In order to make them potent and to penetrate and to bind receptors, usually water solubility is something that we sacrifice. So we get a lot of BCS2 and BCS4 types of drugs discovered. And of course, there are a bunch of solubilization techniques, and I know cyclodextrin is only one of them. We'll not really compare the different technologies. All have advantages and disadvantages. What I really like about cyclodextrins is that usually they combine different advantages in a simple solution. So as I said, they can improve the solubility and the wettability and mask the taste and stabilize the compound all at once. And this is also something that's often exploited, but maybe that's the topic of a different presentation. They're also very potent, but there are also some drawbacks, like they need to be used in fairly high mass, in a high concentration, so that the effect is exploited. So it's always a trade, but we are not pretending here that this is the only solution to solve problems. So let's look at some of the advantages first. So how do we overcome, or how much cyclodextrin can improve the wettability? Because that's one key technical parameter. And in this paper, it's fairly well described and even well reflected how the different types of cyclodextrins can affect the wettability of certain active ingredients. So you can see that the differences can be rather dramatic. So it's very critical to choose the most proper one in order to have the fastest dissolution, not just a complete dissolution. In this video, this is fairly nicely demonstrated. So you can see that on the left, there is the pure active ingredient. In the middle, you can see a physical mixture. You can see that it's dissolving because there is some complexation, and cyclodextrin helps the wettability. But in the right, you can see that the true complex on the very same time frame has a very clear advantage over the other two solutions. And of course, even the physical mixture is not comparable to what you can achieve with the active ingredient itself. So it's a very nice demonstration that was recorded in our lab by the formulation team last week. So what else can we? There are formulations where you can protect the API from light, from heat, from oxidation, even from hydrolysis, or pH for sure, even from hydrolysis to a certain extent. When we're talking about stability, it should be noted that cyclodextrin can protect the active ingredient from all these effects as long as the active ingredient is in the cyclodextrin cavity, which typically happens in the formulation. So, okay, if you make a solid when it's still a tablet, or if you create a lyophilized powder in the vial, even if you create a solution, an eye drop, when it's in its liquid form in the container, typically upon administration, the equilibrium that we were talking about earlier shifts towards the dissociation. It always shifts. It's just a matter of how quickly and to what extent it shifts towards the dissociation. It can be any, and if it dissociates in the body, there will be a lot of competition, a lot of other agents and molecules around, a lot of much larger media. So the complex reformation of the complex doesn't really happen anymore. I'm just saying this, that if you want to protect your active ingredients from an enzyme, that will be troublesome with the cyclodextrin. I'm not saying it's impossible, but it's not something that comes trivial with cyclodextrin applications. So keep that in mind. how do we tune typically a cyclodextrin product when we look at the process? So first, we always start with defining the goal and what kind of limitations are there. So we already said that there are 7 cyclodextrins. Okay, pharma speaking, if non-pharma, narrower range. 7 cyclodextrins that you can choose from, but depending on the the structure of your active ingredient, the administration route, the planned final dosage form, this list may get narrower. So, okay, we understand the project and we understand the limitations. The limitations usually come uh, from a regulatory perspective because not all cyclodextrins are approved for any kind of administration route. Then, okay, we select what kind of product we want to develop, and that would determine what kind of technology we can use to create the cyclodextrin complex. If we can use solvents, again, what type of cyclodextrins can come into the picture, and we do some experiments to select the ones the one or ones that have the best potential with the active ingredient. There could be that different cyclodextrins perform similarly or equally. In those cases, there comes the price, the availability, safety considerations, reference products, whatever. So, but there is a selection, which is typically a fairly simple experiment that you can perform. Then there are certain techniques you can use to improve, let's put it like that, the performance of the cyclodextrins, or you can influence the equilibrium if you want to get back to that, so that it shifts towards the complex formation. This is, I don't know, the concentration of the cyclodextrin, the pH, further components, very important. We'll talk about that, and so on, a few factors. And then if you manage to create a proper product, and the end, you have to make sure that the complexation did happen, what it looks like, how the active ingredient is released from the cavity, how stable it is, and so on. So, without going into details, there are a few limitations that are the drawbacks of using cyclodextrins. These are mainly listed here. No, sorry. These are not the drawbacks. That's another slide. So again, this is about defining what kind of formulation you would be looking for. But this one we tackled so far. I think this is the one with the limitations, and I'm not going into too much detail. But okay, one limitation orally. If you want to develop a generic product, so picture that the originator product doesn't contain a cyclodextrin, and you want to develop a generic that does contain a cyclodextrin for IP or whatever uses. If you're not going into parenterals, this will be very difficult because, as we discussed, the cyclodextrin will very likely influence the solubility, the um, the onset, so the release from the formulation of the active ingredient, and accordingly the pharmacokinetics will be probably modified, and it will be very difficult to develop the exact same product. In fact, and if anyone from the audience knows better, we tried to search for it, but we don't know about a single product where a generic form was developed with cyclodextrins when the original form didn't contain a cyclodextrins. Of course, there are generics where the originator already contained a cyclodextrin, and you can copy and fine tune that one. But typically, the follow-up formulations with known active ingredients are new ones with a new delivery route or whatever. Also, cyclodextrins are very often used for preclinical, early phase clinical studies, but don't end up in the final formulation. Again, another limitation. So if you want fast onset and immediate release, cyclodextrin is super. If you want extended release or controlled release or targeted delivery, cyclodextrin on its own will not work well. This is something that we would like to talk more about in the next webinar, these kind of limitations. And the in vivo stabilization, I already mentioned that at the least challenging. So selecting the proper cyclodextrin, we usually do it with actual experiments. So we typically screen the cyclodextrin and determine how much, their effect on solubilization or stabilization or even taste masking. So we do an actual study, but there are also some predictions and even AI supported tools that you can make. This one is a very nice one. Developed by colleagues from Lille, and it's a good tool to determine not the performance of the cyclodextrin, but the strength of the complex, and it can also suggest what may be the best cyclodextrin of your active ingredient. So it's a nice tool to start planning the experiments with. With respect to binding strength, we can distinguish between weak complexes, moderate, good, and strong complexes. I'm not really saying that you would need good or strong complexes necessary in order for a formulation to work. So there are examples in this area as well. But for sure, if the stronger your complex is, typically, or as a rule of thumb, the less cyclodextrin you have to use in order to solubilize your drug, so your chances are better and better. As this is just a suggestion that, okay, if you want to play a game, try to find as high as a stability constant with a chosen API and cyclodextrins in this platform. This can be fun with students or so. What kind of phenomenons are we talking about when we are trying to evaluate the best cyclodextrin? So technically, what we do is that we increase the cyclodextrin complexation, of course, not in the same solution, but different solutions, and explore its effect on the solubilized drug concentration. In the majority of the cases, we observe this kind of A-type curves that you can see here, especially with modified cyclodextrin. So more or less, as you increase the cyclodextrin concentration, the drug concentration also increases. This can a bit deviate from the linear, maybe because of the stoichiometry. That could be a reason why it deviates. But generally, okay, as you increase the cyclodextrin complexation, you can dissolve the more drug. With native cyclodextrins, in some cases, it occurs that the complex itself has a cap in its solubility, or it's not water soluble at all. And that also have certain uses, by the way. So I don't know, to remove material with the help of cyclodextrin from a media, but that's for another day. Typically, these are the phenomena that you can observe. And then, of course, you select a technique for the complexation. So you made a complex, or you have the right cyclodextrin to make a complex. Now you want to isolate the complex. Okay, you can keep it in liquid, but that's not what we are talking about. If you want to isolate the complex, that's the topic of today's presentation. There are myriads of techniques for that. You can start from liquids, you can start from semi-solids, you can start from solids. We'll see techniques for all kinds of that. Basically, we can distinguish more or less among three methods. So the easiest solution is if you can make, or if you're allowed to make, a common solution with the active ingredient and the cyclodextrin. So technically, you start from the solution of the cyclodextrin. This can be of different concentration, different media, whatever. And you add the active ingredient either proportionally or all at once. You stir, and thermodynamically, the complexes will be formed as the active ingredient would prefer to be in the cyclodextrin cavity. So eventually, you end up in a clear solution. And depending, of course, the concentration of this solution, there are different techniques that you can use to isolate the complex, which can be, okay, this is lyophilization, but other techniques. It's not even necessary that the complex is a clear solution, so suspension would also do with this technique. There are cases where the active ingredient, typically in cases when the active ingredient is very water sensitive, that you don't really want to make an aqueous solution with an active ingredient. In this technique, the co-precipitation or co-evaporation method is something that you could use. So technically, you mix um, the organic solution of the active ingredient with the aqueous solution of the cyclodextrin, and evaporate or precipitate the resulting complexes. If you are not allowed or you don't want to make this kind of liquid-based complexation, there are tools for semi-solids or solid base that we'll go into. Kneading on the lab scale is one of the most convenient ones. So technically, with a physical force, there is a little liquid phase added, but what you usually or what you technically use is a physical force in order to make the active ingredients enter the cyclodextrin cavity. This is also that works very well. So let's see what kind of formulation techniques we have, what we can select from. As I said, in the easiest case is when you can start from an aqueous solution, and that's usually something that's convenient for most of the dosage forms. In this case, okay, you start with creating a buffer. Cyclodextrins are compatible with most types of buffers. I would suggest avoiding very acidic conditions, but even that can be tolerated up to a point. Of course, cyclodextrin do not have any buffering effect on their own. So if you want a stable pH, you have to add something. Then you add the cyclodextrin. Of course, you can solubilize most cyclodextrins to a very high extent, but okay, each have more or less a limit solubility that you can go. As you saw, if you are just running a screening experiment, the higher you go with the cyclodextrin concentration, the higher active ingredient you can solubilize. So for the initial screening, maybe the highest as possible is a good choice. You can heat them mildly so that it's dissolving a bit quicker. And then you add the API, and then you stir rather vigorously and for a fairly long time. So when you are going for the equilibrium, and that's not necessarily the composition that you'll see in the final product, but when you're trying to evaluate the different cyclodextrins, the steering, I would suggest, has to take at least 8 hours, but okay, ideally even maybe 24, so that you make sure that there is an equilibrium. And there are other factors that can play a role, which are described here: temperature, pH, molar ratio of the cyclodextrin and the active ingredient, the stirring time, the speed, so that you put into the consideration, and you come up with a complex eventually. When the other technique, okay, about the isolation we'll talk later, but this is how the complex is already formed. In this case, the co-precipitation method is the other key technique that you could use. So in this case, we have separately the aqueous-based cyclodextrin solution and the active ingredient solubilized in some organic solvents. Ideally, something that's miscible with cyclodextrins, and that's already a limiting step. So cyclodextrins are not very well soluble in most organic solvents. Some cyclodextrins that are at least approved for pharmaceutical uses are not soluble in any kind of organic solvents. Okay, methanol and ethanol are borderline or to some extent work. You mix and stir these solutions, and okay, after cooling, you remove the solvent, or if you have a precipitation, you do centrifugation, and then comes the drying. So as I said, okay, making the complex this way is not more critical either. It's especially good if you it can be useful also to incorporate a higher amount of the active ingredient into the cyclodextrin. It's a bit more difficult to do it this way, but it can be useful for that. And in case the active ingredient is sensitive to hydrolysis or water. Then moving slowly from the aqueous based techniques to the semi-solid and solid based techniques. So when you're doing a grinding, you prepare kind of a paste or a powder, okay, a very dense paste, let's put it like that. And of course, when you're doing the the milling, it's more like a powder, but process wise they're all the same. In this case, the driving force is not really the thermodynamics of the active ingredient, or not only the thermodynamics of the active ingredient, but also the physical force uh, that's making sure that the interaction between the cyclodextrin and the active ingredient happens, which in aqueous phase would be um, more straightforward. Here you need an extra force. As you can see from here, and we are rushing a bit ahead, but okay, you can use different techniques to check out if you could form a complex with the with the active ingredient. And theoretically, when you look at the spectrum of the active ingredient and of the complex, the in an X-ray, for example, the original crystallinity characteristics of the crystallinity of the actives should completely disappear, and only the amorphous nature of the cyclodextrin and of the complex should be shown. And with these kind of techniques, you cannot always achieve 100% complexation. So these are to achieve a perfect complex with especially solid phase complexation is much more challenging, and you have to fine tune a lot more the conditions than compared to the solution state. Kneading is something that's okay, more like okay. So co-grinding was a solid form. This is more like a semi-solid form, and industrially it can be translated to extrusion. It's a very efficient way to make a complex. Is also fairly high amounts under lab or kitchen, let's put it like that, circumstances, so you can create a fairly high amount of complex with this technology. Here the kneading time, of course, and the amount of liquid or solvent that you add are very critical, and it's of course not easy to implement some kind of in-process control to make sure that the complexation is actually happening. But in these cases, this is something that I would highly advise if I were developing a product either with the milling or with this kind of extrusion technique. I would certainly suggest to make sure with some in-process control that the complexation happened to the extent it was planned, and there is a consistency batch to batch, because unlike when you make a common solution, in such cases there could be a lot of differences from equipment to equipment, and from technique to technique. So how do we isolate the complexes from the liquid phase? Spray drying, I would say, if you're not talking about parenterals, that's probably the most widely used. So both in the nutraceutical area, or if you just want to isolate the complex as an intermediate ingredient, or if it's already a, I don't know, a final product for capsule filling. Spray drying is one of the best ways. You can create a fairly high, fairly concentrated solution of the complex, and well, yeah, you can spray dry it, and it's probably the easiest and that has the highest capacity to create a nice complex. Freeze drying is also one of the most popular ones, especially in the realm of parenteral formulations. So most of the products that are uh, injections with cyclodextrins are marketed as a lyophilized powder for reconstitution. Again, works very well also in the lab. We use it all the time. You can tune very nicely the different properties of the freeze drying process to ensure the proper final characteristics of the product. So again, something that I would certainly recommend for complex isolation. Of course, at the end of the day, it's it's technically the final use and the characteristics of the active ingredient that puts a lot of limitations around which technique we select. So, okay, if your API is heat sensitive, you cannot choose spray drying, irrespective of what you want to achieve in the end. So you have to settle with I don't know, or precipitation or spray drying or freeze drying. Okay, it depends on what kind of dosage form you will need. You will need a liquid, you will need a solid. Do you want to process it further into, I don't know, making granules because you want to make a tablet? In that case, of course, you will not go with with freeze drying. I really just wanted to show you that there are a bunch of techniques available, and probably more beyond that. I just wanted to show the most frequently used ones that you can use to isolate the complex. But at the end of the day, it's the characteristics of the active and the further processing which would determine which one you have to choose, and of course what's available at your side, because of course that's the most trivial limiting factor eventually. So you have a complex now. There is one, and then you know how to create a complex. Now there is one part which we haven't talked about. We talked about okay, how you can play with the pH and temperature and cyclodextrin type and concentration to improve the performance. But let's take a moment to talk about cert components, because these additives for non-binary formulations can help a lot to boost the performance of cyclodextrins. And honestly, if you look at the products that are on the market, in most cases they do contain something else uh, apart from the cyclodextrins that's supporting the cyclodextrin complexation, either in a way that it's stabilizing the complex, or either in a way that it's contributing to the formation of the complex. Usually, these are some kind of acids, either hydroxy acids or amino acids or polymers. So you can only, or you should only think about, okay, these are just as common excipients as cyclodextrins, but they have a synergistic effect. That we always or often see. Unfortunately, this kind of synergistic effect is non-predictable. So as we cannot really predict which is the good cyclodextrin that would work for your active ingredient, we will not be able to predict what kind of cert components to use. So you have to evaluate them, and it would probably differ from cycle also on the cyclodextrin itself, um, not just the active ingredient. But to show you an example why it's important to use it, so you want to dissolve domperidone without the cyclodextrin or without any active. 10 milligrams takes 2 liters of water. If you dissolve it in cyclodextrin, 62 milliliters. If you dissolve it in hydroxy acid, it's 12 milliliters. But if you combine in aqueous media, of course, the cyclodextrin and the hydroxy acid, you're down to 0.3 milliliters, because in this case, I think it was citric acid in this case, I'm not entirely sure, maybe tartaric, the cyclodextrin have a synergistic effect. And we can see similar examples. Another one is what kind of form of the active ingredient we use. Surprisingly, it would have a dramatic influence. You can imagine that depending if you use the free base or the salt, would have an effect, because the intrinsic solubility of the free base and the salt would differ dramatically. Of course it does, but and then also the different types of salts has a very different solubility. But not just that, but how much the cyclodextrin improves the solubility of the different salt forms. So here you can see that, I don't know, between the hydrochloride and the tartarate, the intrinsic solubility is okay. There is a twofold difference, but in the cyclodextrin complex, the difference will be always almost 6 or 7 folds already. Whereas similarly, I don't know, the mesylate that had a very good intrinsic solubility was not that much improved compared to these ones with the cyclodextrin. So the type of the salt form or the active ingredient, or the what type of form you use is also very critical in the complexation. Just wanted to emphasize that. This one is about the effect of polymers. It's a very nice technology where it's demonstrated that how much the use of a polymer can improve the performance of the cyclodextrin. So here you can see, and of course, don't really focus on the native form, focus on how much it improved the complexation, or the concentration of the curcumin compared to the cyclodextrin form. And I'm not saying that it's guaranteed that this kind of dramatic improvement can be achieved, but for sure, similarly to hydroxy acids or amino acids, certain types of polymers can have this kind of dramatic effect, both on the release profile or the total concentration of the active ingredient. So now you think that you created a complex, so how you can see if you actually created a complex? In general, we could say that any analytical method that can distinguish between the free active ingredient and the complex active ingredient is suitable to detect the complex. So I don't know, just imagine an HPLC. If you inject the naked active ingredient, it would have an elution time. And as you can imagine, if it's captured in a cyclodextrin completely, then this complex would have a different elution time because it would bind differently to the column. What happens in reality is, of course, that not all your actives are in a cyclodextrin cavity. So I don't know if your peak naked active is here, the fully complex is here, and you inject a solution that's a mix between the two. I mean that not all the actives are in the cyclodextrin complex. Then you will see a peak in the middle. You cannot see distinguished the free active and the complex active and say that, okay, I don't know, 25% of my active is still free. But you can see how much it's shifted from the free towards a hypothetical full complexation, and you can calculate the complexation rate. And this you can do with all these measurements that are or all these kind of analytical techniques that are on the table. Just what I mentioned before, about okay, how we detect that there is a complex. The most straightforward ones are shown on this DSC or X-ray. The original properties of the active ingredient, the structures or the melting points disappear because they became complex, and they properties change for the time that they are in their complex form. So when there's a full complexation, this is what you should see. And of course, there is also microscopical techniques that you can use. Again, just a few further examples about, okay, it's not just the complexation itself, but assessment of the stability of the complexes that's also important. Physical, thermal, chemical stability, that's a very key criteria. You have to make sure that the active ingredient stays in the complex, not just at the time of the making, but over the storage time, and it can protect the active ingredient. You can also assess the binding strength. As I said, in my opinion, it's not very critical how strong your binding is. And my experience also says that you can achieve nice products with weak binding too. But it's something to help certainly understand, and it's not big of a trouble. And again, there are a wide variety of techniques that you can use to assess the binding strength. You can also assess the stoichiometry. So what is the molar ratio of the cyclodextrin and the active ingredient in your final formulation? Here, what you should know is that this is really true on a molecular level. So if you determine that your complex is a one-to-one complex, that will be true on a molecular level, but that doesn't mean that you have to mix one molecule of cyclodextrin. Okay, so on a molar level, 1 cyclodextrin and guest molecule, and in that case, all the guest molecules will be incorporated in the cyclodextrin cavity. And that's because we are again talking about an equilibrium. So your guest compound will exit the cavity of the cyclodextrin, but you want it to find the cyclodextrin, an empty cyclodextrin, as soon as possible so that it forms a complex again. And this is the easiest to achieve if there are empty cyclodextrins in the system. So you have to use extra free cyclodextrin, an excess of cyclodextrin in these formulations. How much the excess? Well, that depends how much the guest molecule wants to leave the cyclodextrin cavity. If it doesn't really want to leave the cyclodextrin cavity and it's happy in the complex form, then just a pinch of extra would do. There are products on the market which has, I don't know, 1 to 1.4 as ratio of the active and the cyclodextrin. So I don't know, 40% excess of the cyclodextrin. There are products, okay, I don't want to cause you nightmares, but there are products that have molar ratios of to one active ingredient, there are 15, 17 cyclodextrins in the system. Well, okay. So theory, reality. Then maybe the most important among all is the geometrical assessment. So how your active ingredient fits the cyclodextrin. We have two entrances. They are not identical. So one is narrower, one is wider. Different parts of the molecule can enter into the cyclodextrin cavity, which would have an influence on how well it stabilizes it, how well it solubilizes it. And this is also something that in the pharma field often asked by the regulatory. So that you explain that, is it really a complex? How it's fitting into the cyclodextrin? What happens? How is it releasing? So with such kind of two-dimensional animal studies, you can create very nice visuals, or with computational modeling. That's also a nice tool to do it. And of course, now you know that you have a complex. Now you know what it looks like in reality, and you have to make sure how it dissociates. So how the active ingredient gets released from the complex. What is the kinetics of that, how it if this kind of complexation helps membrane penetration, if it affects protein binding, whatever. These are all possible with the help of just common analytical tools. So this is technically what I wanted to capture today. I hope it was understandable and could provide you with some extra insights into the world of cyclodextrins and cyclodextrin complexation. As a take-home message, what I would say, it's very important to pick the best cyclodextrin. It's not difficult to do, but I would always do an actual real lab trial and do not rely on any kind of assumptions, because there could be surprises. Then to fine-tune the complex with further components in order to have an optimal composition that will not only contain most probably cyclodextrin and the active ingredient, and find a proper technique where you can isolate how you can isolate the complex that's fitting to your further goals and make sure that the complexation did occur as you expected. So the final product, when you isolate it, is actually a complex, not just a mixture of the cyclodextrin and the active ingredient. So, thank you for your attention. Oh, I was talking way too long. That's not good, but okay. Oh, okay, let's save some time. Um, so we reserve some time. We'll not close the session at 5. I'll try to answer as many questions as possible. So just type them in the chat box and I'll scroll them. Okay, the guesses were quite wild. 100 cyclodextrins. Major 9. Okay, someone was doing very good. So do cyclodextrin API co-solutions with DMSO or buffer work? So most cyclodextrins are not soluble. At least the cyclodextrins that are approved for pharmaceutical uses are not soluble in in DMSO. So it's not working that well. If it's soluble, it can work. But okay, that wouldn't be my primary choice. Are there data, I mean complex stability data, Anna is asking, for complexes anti-tuberculosis drug ethambutol? Okay, so I don't want to start Googling, Anna. Of course, I don't know. All the products are out there. If we are talking about very old and well-known active ingredients, I would say that it's really rare that we don't find any kind of evidence or prior art with cyclodextrins. So if you can complex ethambutol into cyclodextrins, I'm sure that there are papers about it. I'm not sure which one are those and what's the stability constant itself, but okay, a quick search, but probably do the deed. How does the choice of cyclodextrin type, alpha, beta, gamma, or modified, influence the solubility and the stability of poorly water-soluble drugs? Okay, so I can, of course, only answer Irfan's question on a very high level. So, typically, modified cyclodextrins like the hydroxypropyl beta, the methyl beta, or the sulfobutyl beta have a much better solubilizing power and stabilizing power than the native cyclodextrin. So than the alpha or beta or the gamma cyclodextrin. That's partially because their water solubility is unlimited. So you can create very high concentration cyclodextrin solutions. And as we have seen with these cyclodextrins, there is almost always a linear interaction. So the more cyclodextrin you add to the solution, the better solubilizing power you will see. And also because of their side chains, they are capable of forming extra interactions. So when you imagine there's a cyclodextrin and we have an active ingredient, okay, and always it's like, okay, part of the active ingredient is in the cyclodextrin, but part is sticking out. I'm sorry. And a part of the active ingredient is sticking out. And when you have a native cyclodextrin, then the part that's sticking out cannot interact with the cyclodextrin. There's nothing to interact with. It will just be there. But when you have a derivatized cyclodextrin, you have side chains like a jellyfish on the surface of the cyclodextrin that can still interact with this outer part, forming hydrogen bonds and secondary interactions in case of the sulfobutyl ionic interactions and so on. So the complex is stronger, and that's why the solubilizing power and usually the stabilizing power is better with the derivatized ones. Not always, but okay, majority of the cases. So if the project allows, and so for nutraceuticals or no, I'm not saying that. For nutraceuticals or food products, you are limited to the native ones, alpha, beta, and gamma. You cannot use anything else. In the pharma, you have more freedom, or in the cosmetic field, you have more freedom, and that's why in those fields, usually they do use other derivatives because they perform better and are not that much more expensive, especially nowadays. Then, David's question, when we obtain dry complexes for lyophilization, are the complexes still stable even when I redissolve them again? Oh, this is a very good question. So, you isolate the complex in the lyophilized form, and then you freeze the complex. So in that case, of course, you freeze all the circumstances. Most of the actives will be in the cyclodextrin cavity, and in the solid state, it will stay like that for a very, very long time. I mean, talking about years, maybe tens of years. We have done extremely long stability studies in the past to show that complexes still exist after a very long time. When you redissolve, if you make a concentrated solution, so if you reconstitute to the extent of the media that you used for the lyophilization, the complexes will be just like they were before. But okay, like talking about a vial again, you want to make an injection, and you lyophilized the injection into the vial, and you reconstitute in the vial, you shake, everything is dissolved, all the active ingredients will be the complex. You put them in the syringe, inject, and the complex will dissociate upon dilution after the injection. But as long as it's in the vial, it will still be that way. It's just more stable, and shelf life is typically better in the solid form, of course. Then the next one. Okay, Anna, is it possible to obtain the slides? So if you write me, I send them over. Okay. Possible to identify the 9 cyclodextrins. Okay, so the 9 cyclodextrins were alpha, beta, gamma. I missed the slide about that because, okay, that's not what I really wanted to talk about. Alpha, beta, gamma, so the native ones. Hydroxypropyl beta, hydroxypropyl gamma, sulfabutyl beta, and methylated beta. These are the ones that you can use as excipients, not unlimitedly, but as those. The cyclodextrin for vaccines is called sulfolipocyclodextrin, but it's very rare. It's only a component of one single product, so it's not something that's widespread to be used. And Sugamadex is the active ingredient. Yes, they are in equilibrium. Very interesting presentation. One question about the regulatory status of the cyclodextrins. Very few are included in the inactive ingredient database of the FDA. Is it difficult to register IV products that contain cyclodextrins that aren't in this database? Oh, nice question. So, by the way, I think not the next, but the one, the Masterclass 7 will be about basic characteristics of the cyclodextrins when we'll assess safety. But yeah, I know that in the active ingredient database, there is, compared to the 7 that I mentioned, surprisingly low amount of cyclodextrins presented. So I don't know, for orals, there would be only the beta and the hydroxypropyl beta, whereas there are other products with other cyclodextrins too. For IV, there would be the hydroxypropyl beta and the sulfabutyl beta, which is okay, covering the majority of the uses, but there are other products with other cyclodextrins for parenteral applications out there. In my experience with regulatory discussions, when you use ones that are listed in this register, you don't really see troubles. That are not in the register have some products on the market, but there are not too many references. So I don't know. There are parenteral products with hydroxypropyl gamma cyclodextrin, maybe one or two, compared to, I don't know, the 20 with the hydroxypropyl beta and the sulfabutyl. So the references are much less that you can use. Also, the dose range of the safety profile is much narrower. So the available data is not present to that extent. So, in the sense that it will trigger further questions from the regulatories and probably the necessity of the further studies, I would say that it's a likely yes, that it would be a bit more difficult to use them, or use other types. You would have references, but probably you would be asked to do more studies than with very common cyclodextrins. Okay, in GCMS, how do you prepare the sample and what hallmarks tell you that you have formed the complex? Okay, so I'm not an expert in GCMS. I can, Brenda, I'll make a note to myself so that I send you some papers about it. It would be a bit too long to explain it, how it works in this particular application, but it's feasible to analyze the complex with that technique too. What would be the easiest way analytical methods to confirm the stoichiometry? Um, if your compound is UV active, then a UV, so stoichiometry determination works in a way that you create a stock solution from the active ingredient, you create a stock solution of the cyclodextrin, and you mix them in different ratios. These stock solutions, and that would be on the one axis, the ratio of proportional ratio of one of the components, and the difference in the shift will be on the y-axis. If the active ingredient is UV active, that's the fastest. Usually NMR is used because it's more universal, not as closely related to the active, but similarly to determining stoichiometry, any other technique would work. UV is the simplest, I think. Then, okay, just compliments, Darius. How can we prove the formation of an inclusion complex with SBCD, which is randomly substituted? Due to its amorphous nature, it's possible to obtain a single crystal of a complex and determine its structure using X-ray. Moreover, rosy spectra obtained with NMR are strongly affected by the presence of artifacts. Could you kindly suggest another way of demonstrating that true inclusion takes place? So, with these cyclodextrins, yeah, they are amorphous. Also, the complex will be amorphous. Making a single crystal of the complex is extremely difficult, even if the cyclodextrin is not a mixture like the SBCD. If it's a mixture, well, like SBCD, I would say impossible. Still, I think X-ray should be okay. So I understand maybe your problem is that it's an indirect proof, um, that okay, when you compare the X-ray of the active ingredient to the X-ray of the complex, what you see is not the complexation taking place, but that the original signals of the active ingredients disappear, which is an indirect sign. With the rosy artifacts, I would argue with that. That's works very well to see the geometry and the inclusion of the active ingredient. So if you want direct proof, rosy is probably the best one that you can use. At least that's the one that we almost all use in almost all the cases. Yeah. Okay. You said that active ingredient signals could overlap with cyclodextrin signals. That's true. That's true. Okay, in that case, IPC is also working quite universally. ITC, sorry, as a thermal titration colorimetry. That's also something that usually works very well. Maybe that would be my suggestion. Arcana. Um the problem that I faced working with cyclodextrin is to analyze the complex tissue using NMRs. The proton signals are always overpowering the gas proton signals. Yes, that's of course because you are using much more cyclodextrin, I mean molar wise, than than the active ingredient, and the active ingredient signals are very small. Try ITC, it works fairly well. But I see that there are other suggestions too. I know. I like it that you the discussions that you entered into. Okay. We still have. Could you explain the advantages of BCD in soft capsule formulations for hydrophobic drugs such as paracetamol? Okay, Charbel. So the next masterclass will be about oral formulations. So if you don't mind, I'll tackle that question next next time when we're talking about oral formulation specifically. In the co-precipitation method, is there a risk that an applied nonpolar solvent would compete with a gas molecule entering the cyclodextrin cavity? Um, yes and no. Depends a lot, Darius, on the type of the solvent. So with methanol and ethanol, alpha cyclodextrin, not the bigger ones, alpha cyclodextrin can form a complex which is not really strong. So if you have a hydrophobic active, I wouldn't say this is a real risk, but it's a valid point that this is more competitive than water. For sure. Usually we don't see it as a problem. It can be seen as a problem when you want to get rid of the solvent, when the question is if you can reach, regulatory or pharmacopoeial standards, and the empty cyclodextrin. So I wouldn't say it as a risk that you cannot encapsulate your active ingredient. They will find cyclodextrins. The problems is that the organic solvent may be encapsulated in the cavity of cyclodextrins, and during the drying process, it could be very difficult to get rid of that part of the organic solvent. So if you use something that's toxic or have low safety limits, you will be in trouble. That's why we usually use ones that have higher limits, like ethanol. Mhm. Could you suggest a method to prove the formation of the complex in solid state sample without the need to dissolve it if the powder is fully amorphous? I would still, well, my best suggestion would be the X-ray or the DSC to compare it with the active ingredient. And I hope, okay, at the end of the questions, if you have further questions, I hope that you could get our email, or okay, you can find it fairly easily. Feel free to drop a line to us. And I'm really happy that so many of you enjoyed the presentation. We'll have another session, as I mentioned, about oral formulation specifically in about a month's time. So if you follow us, you'll see where to register. Thank you, everyone, for the nice session and the very interactive discussion. I hope everyone is happy. Have a good evening. Bye bye.